controlled, various activities that are controlled by the action of molecules upon molecules. So all this can be identified using these network representations. So again, these are the kinds of terminologies that are present when we will look for any kind of network analysis. So paths, the path between these nodes, these are nodes, these are edges, and this is the paths they follow. So if I'm talking about this node is regulating this next node, which is getting regulated by the third node, which is in turn associated with the fourth node, which is in turn regulating the fifth node. So this becomes the path from here to here if I am reading this. Cliques are protein complexes. So if a complex is linked together like this one, so this becomes a protein complex. Or regulatory modules are again small modules where there is a regulation happening. So here you can see this looks like a regulatory module. Everybody, this is regulating all the other nodes in the network. So this is regulating this protein, if this is a protein interaction network. So this node is regulating 1, 2, 3, and 4 proteins or genes, whatever they are called for, and they can be looked upon like this. And subgraphs are those which are maximally weighted. Weighted means that it has maximum number of interactions associated with it. So those will become the subgraphs. So so let's move on to another kind of information that is important for us when we are doing network studies or network analysis. So the first and foremost thing is to understand
which
what
are
kind of,
maximum.
sorry, hello, am I audible?
Yes, madam, you're audible.
Okay, so I think there was somebody saying thank you. Okay, thank you so much. All right, I'll go full screen then. Okay, so I was explaining about the directed and the undirected networks. And so this is another very important point to understand when we are working with network analysis, that what kind of network we are working on. So just to look at this, I think it makes our life clear that what are the terminologies I'm talking about. A directed network is one which has directed edges. So A is regulating B, B is regulating D, D is regulating E, and all the edges have angles to it. So they have a direction. So this is a directed network. And when the network has either it has all nodes that are undirected, or it has some nodes that are directed but some nodes that are undirected. So Cytoscape in that case will consider this as an undirected network only. So just to make it clear, the directed network contains solely directed edges. It provides 3 possible interpretations of edge direction, and you have to select the possible interpretation for further processing of the network. Undirected network has both undirected and directed edges. So undirected edges cannot be converted unambiguously to the directed ones. So mixed networks or mixed edges are considered to be as undirected ones only, because we cannot decide it that okay, let me put an arrow, let me put a dash against it. This has to be coming experimentally or with proper validation only. So this is what you see. If you see a network, this is how network looks like in Cytoscape. So interpretation 1, this network is directly directed. Interpretation 2, network is undirected. Edge directions should be ignored in this case. The only thing that is available as an interpretation is that these three are connected to each other, but they are protein-protein interactions or simple interaction or a complex, but we do not know which is regulating what. And this is the third representation where edges A to B and B to A represent a single undirected edge, that is A to B, because there is there is not a feedback loop is not working there. So this is not they are not regulating each other. A is regulating B and then B is regulating A, that is represented by a single undirected edge A to B. So we have a simple network with the number of connected components and parameters related to shortest path and parameters related to node the neighborhoods that we study. In complex networks, we study clustering coefficient, neighborhood connectivity, and degree distribution. So this, for example, node degree, if I talk about, if you see this particular node, it has a high degree because it has a lot of connections associated with it. And if you see this node, it has a low degree because it is connected only to one node that is there, possibly attached to this particular node that is present. So node degree is defined as the number of connections you have. Then you have clustering coefficient. Like again, you can see that there is a high clustering here happening. These are all interconnected to each other. Also, there are paths that are following to the central node. They are also interconnected to each other. So it has a higher clustering coefficient, whereas this one will have a lower clustering coefficient because it is only connected this ways, and it is not connected to the other side, and that is why it is showing a lower clustering coefficient. Then you can find out the path length, path distance, the motifs that are present, or which we call as hubs in a network, or you can find out the common structures, community structures, modules, and hub. So this is module 1, this is module 2, and this becomes your hub gene that is common to both module 1 and module 2, will become your common structure or hub gene. So let us understand where the data is coming from, because we cannot be building these networks by ourselves. Either the data is experimentally coming to us, that we have a list of proteins or list of genes with us and we want to make a network from them, or we know that there are repositories that are available which we can work through them and find out the data that we are trying to figure out. So some of these repositories are mentioned there. I'll take you through one by one all these repositories. So the first one is Biosize. Biosize is a genome pathway genome database for organisms with completely sequenced genomes. You can see this is how the home page looks like. This is the home page representation of Biosize. It is present at this location, www.biosize.org. It is an open access database. It has 409 full genomes and pathways that are deposited into it, which are curated. And it has species specific pathways that are inferred from MetaSize, that is the other database that we are going to be talking about. So it has a lot of information. You can see it has 20,023 pathway genome databases information to search for modeled eukaryotes, and for thousands of microbes, and software tools are also available in this particular resource that I have showed you. You can go there and try for free. There is an introduction video also, that introduction or a tutorial also that is available, or that is there, and you can browse it to understand more. And then if you want to go into the details, you have to take the access for or subscribe to it. So query or navigation pathway creation support can be possible through the pathway tool software suites that are present in the Biosize database. So this is a snapshot of all the information. There are 3 tiers. Tier 1 is literature derived information, which is MetaSize and EcoSize. Tier 2 is computational derived databases featuring some curation. There are 13 databases under that category. And also tier 3 has computational derived databases without curation, which has more than 300 databases. So when I say curation, it is basically adding information to any kind of data that is present. So if the data curation is done, there are around 13 databases which has some curation, and more than 300 databases which have only information on these pathways, reactions, and substrate, but will not have any curations associated with it. So the following websites that you see: Human Size, B Subsize, MetaSize, EcoSize, Pathway Tools, and Biosize, these are all part of the Biosize consortium, and they are all listed there under the different headings. Then you have under this only, EcoSize is basically the E. coli encyclopedia. It is a review level model organism database for E. coli, and it tracks evolving annotation of E. coli genome and cellular networks. So there's a comparative genome analysis you can see on the screen, which is available. There are tools that are available which you can be working through and finding out more information on them. It has curated commentary, means information for all experimentally described genes, which is around 3,300. It has 15,000 supporting citations, and both a text and computational reference is available for all these information that you have to portray. Next is MetaSize. So MetaSize is basically a metabolic encyclopedia. It is a non-redundant metabolic pathway database. The goal here is to describe a representative sample of experimentally determined metabolic pathways. It is a curated databases that have the all the elucidated metabolic pathways from all domains of life. It is based on literature-based curation, that is from research papers of pathways, reactions, enzymes, and substrates. It has over 800 pathways from 700 organisms that are present, and it was jointly developed by SRI and Carnegie Institution. It is having a lot of information again is present at the metasites interface. You can find out you can find out a gene, a protein, a metabolite, or a pathway, and once you click there you will get more information on what you are looking for. So, curation is a process, as I told you, of manually refining and updating a bioinformatics database. MetaSize project uses a literature-based curation approach in which the database contents are extracted in a stepwise manner. As you can see the flow chart, it is extracted from a proper repositories, databases, experimental literature, and then the curation process occurs where data extraction, data synthesis, data quality control is done. And finally, you have pathways, you have a list of compounds, you have a list of enzymes and enzyme complexes, reactions, genes, and more. So this is what you can see is the process, and there are other databases such as Arasize, which is for Arabidopsis, Ecosize I just spoke about, yeast biochemical pathways databases, and Scosize, which is for the Streptomyces colicolar organisms. So these are all organism specific databases which are present, where you will get a lot of information based on your organism of interest. So again, this is about metasize only, that they include they have a significance of the pathway. Every information that is captured with a significance of pathway, relationship with other pathways, organism within the pathway that exists, experimental evidence for the pathway, contradictory evidence, and citations. So it has around 2,800 metabolic pathways which are present in 3 tiers, where you can see there is an increasing level of curation. So tier 3 has minimum level of curation, tier 2 has the second, and then comes tier 1, which has the maximum level of curation or information added to it. This is just a snapshot of what glycolysis looks like, and you can click on each of these terminologies and find more information. You can go on less detail, and you will get more idea about each of these metabolites and what is happening. So this is what a pathway will look like when you browse through metasize through it. So next is KEGG pathway database, which is again a very known database, and it is very, very useful to get the information. It is it is called as Kyoto Encyclopedia of Genes and Genomes, and it has a knowledge of molecular interaction networks, about the genes and the proteins, and about chemical compounds and the reactions. So this has a representation of systems information, health information, genomic information, chemical information, and which will have a lot of data that is pertaining of basic principles in natural sciences, practical applications for use in societies, personal genomes, pathogenomes, genomes, metabolomes, and genomes and metabolomes. So all these informations are present in Kyoto Encyclopedia of Genes and Genome, which is an interesting database again. It is an integrated resource which has these categories. You have KEGG pathway, KEGG bright, KEGG module. So once you explore all of them, they will get you a lot of information which will be useful for your research, as well as for understanding the basic information on all these processes that you can build on. So these are the major components. I'll not go there. I will just show you this is how you can access the KEGG pathway. You can click here, click on KEGG pathway, and you can type glycolysis, or it will give you both gluconeogenesis, which is the backwards of this process. So here you see it is the whole representation that is given to you in the format of KEGG. This is the KEGG format that they follow. And these, what you see, 2.7, 5.4.2, these are called as EC numbers. These are enzyme classification numbers. When you click on them, you will get more information about these numbers. And then you can keep on expanding your search and find out what you want for each of these resources. So this will help you in turn to get more idea about each of your metabolite that you're trying to work with, or the enzyme that you're working with, or the substrate that you want, everything related, and it is cross-referenced. So a lot of information you will get from this single resource. So let's come to the next database, that is the biomodels database. But that biomodels database is a repository of computational biomodels of biological processes. It allows the scientific community to source, search, and retrieve mathematical models. It has 86 curated models and 40 models that are pending curation. Probably by the time we go back, we'll get more models that are present. You can browse more information. You can see 640 models are now available, which are manually curated, and more than 1000 models are present which are non-curated. So you have a lot of information, and this slide just gives you how representation in biomodels looks like. So this is taken from the biomodels, that each of these processes can be listed from there, and every database has its own representation. So this is again glycolysis pathways in biomodels. So I've been showing you all the pathways that are present in different databases. So let us just compare all these pathways databases. What I talked about, metasites or biosites, the curation is manual and automated both. KEGG pathways have automated manuation. The KEGG curation is automated. Biomodels again is manual. The size, it is more than 621 plus pathways. It has KEGG has 289 reference pathways, and biomodels have more than 126 models. Nomenclature is EC numbers and gene ontology, EC numbers and KEGG ontology, and gene ontology for biomodels database. Organism coverage is more than 500 species. KEGG pathways has various, and biomodels have more than 475 species. It is a species specific, custom based visualization, reference based, and species specific. Biomodels have a non standard representation. Primary usage is for pathway comparison, computational biology, simulations, and modeling. Then again, the Reactome database is another database which is a curated database of studying interactions. It is the information in the database is authored by expert biologist researchers and maintained by Reactome editorial staff. It is cross-referenced to NCBI, Ensemble, Uniprot, UCSC browser, HapMap, KEGG, CHBI, and GO and PubMed. So Reactome also helps us, and the SBML files that are exported by Reactome do not contain any quantitative information, and it describes the human cellular pathways accurately in full details. So let us now come down to protein-protein interactions. These are physical and selective contacts that happen between the pairs of proteins and in certain molecular regions in a defined biological context. Interactomes is the totality of PPIs that happen in a cell, and all these representations we can go ahead and find out in the Cytoscape. So before I begin the Cytoscape part, I'm just summarizing, and these are more databases that you can be probably listening, finding out more on yourself. In ACT, Mint, and APID, these are more resources where again you will have information on the protein-protein interactions. So now comes this homepage of Cytoscape. Cytoscape is an open source Java based bioinformatics software platform, which is used for visualizing biological networks and analyzing networks with gene expression profiles and other kind of data. It is the core features of Cytoscape are that it has a customized network data display. It has a powerful graph layout tools. Then there are easily organizable multiple networks. You can merge different networks together. You can easily navigate large networks. Then you can filter the network, find out the subnetworks. And then there are plugins or APIs which will help you to analyze the network into more detail. Then the workflow of any cytoscape experiment is that first part you obtain protein-protein interactions. These interactions can come from any of the databases I just spoke to you about. You have to take the information from these databases, curate the information yourself as per your biological question of interest. Then you can visualize the data in terms of your network through Cytoscape. It will build a network in Cytoscape and perform basic network analysis in Cytoscape, identify heavily connected and subnetworks. That is what is the basic thing you do. So to start with, we can just have a small network that can be imported from public databases if we are not familiar with, or if we do not know how to start with a small network. What we can do is we can just open Cytoscape and then click on file. The file option will give you an option of import. The import option will give you an option of import from public databases. So these are the different kinds of resources. You can also take it from a table. You can take it from a table, from a URL table, from a public database. You can take network from a URL. You can take network from a file. And NDEX is kind of an internal import policy that Cytoscape has, which will fetch the data for you, and it will also create the network similar way to the way the string protein query will work. So once you click here, it will open another window for you where you can select the data source. Like in this case, I have selected string protein query. You can select the organism, Homo sapiens, and then either you select all proteins of this species. So whatever is inbuilt in the string database, it will give you all the proteins. If you do not want that it should have all the proteins, you can just paste the protein names or the identifiers. These are UniProt identifiers that you see. So it supports UniProt identifiers. It also supports ensemble identifiers and a whole more list of identifiers. You can check that on the data resource as per your information, whatever is present. And once you paste the gene symbols, when you click on import, here at the end of it is a click button called import. It will import this from the string protein query database, and it will give you the kind of network that you see here. Okay, so this is the next output that you see on the screen once you import the network. So imported network can be, it will be coming by default as it is present on string. Even if you run the same thing on string, there is an option that it will be directly present like this only. Okay, so once you have the network, you can do a lot of things. Again, you can change the layout. That is the most basic thing that you do. When you click on layout, there will be a lot of options that are available where you can change the layout, and now the network looks like this. So these are some of the very basic features that you can be working with the network once you open them. So then you have a lot of plugins. Here I am showing you just one plugin that is available, that is mcode. Now mcode is used to find out the modules from the, or the clusters in your network. That is the information that is available, that is the most hub genes which you call. So for apps, you need to install the apps through the app manager, and then you have to select the app that you're working on, select the options. So these options are selected by you as the user according to what is your interest, and then you will have the output which you can analyze by getting more information on the network clusters. You can click them and more information will be floated through them. So you can select sub networks like this. You can just click on this. Once you click on this, this will open up. This is the network that this is the first 86 nodes and 765 edges. This is the first step sub network that it could find out. And from that, if you can select a particular portion, you can select the nodes of the sub network and then find out more information by clicking each of them and analyzing them in details as per your biological question. So this is what I have. This is what M-code highly connected network will look like. And then you can change the nodes here. If you click on these nodes, you can change the colors, you can change the representations. If you want to change it, you don't want it like an oval, it will change to a square. If you don't want a square, you can change it to whatever representation you want. You can also change the edge sizes. You can make some more thick, some less thick according to your interest, and you can also be working through it in more details. You can start working through them depending upon that this is my interest gene. You can take it out, do further. So why did we take this session? Network analysis has a lot of information. This was just a snapshot of how Cytoscape can be used. There are a whole lot of functionalities and many things that you can do with Cytoscape with your data actually. So what are the basic outcomes, or why is it so useful? It will give you the gene function prediction. It will help you identify the connections to the set of genes or proteins involved in the same biological processes. It will give you the statistically significant domain-domain correlations in a protein interaction network. It will give you common pathways that are affected by a collection of genotypes. You can do network-based diagnosis or biomarker selection. You can also identify a disease network, subnetworks that are transcriptionally active in a disease by combining some expression data into your network, which will be useful for you. So this is all about Cytoscape from my end. As a beginning, Cytoscape in itself is a whole tool which has a lot of functionalities, and there are a lot of things that you can do. But a basic representation how you can fetch a network, this is one way of doing it. The second way of doing it is through Cytoscape itself. So I will just show you the Cytoscape window, and you can just open, click new network, empty network, and whatever name you want to give. This is the network you have. Now you can create add node, and a node is here. You can change the edit, rename node, it becomes A. You can create another node and this becomes B. So like that, if you want to have so this is how you can just create a sample network by yourself. I am not fetching this network from anywhere. I am just creating this network myself. Then I can rotate it around. I can edit it. I can group it. I can select the first neighbors that are undirected, use apps to extend by public interaction, and so on and so forth. So this is just a basic overview. Obviously, it is not beyond the scope of the time that we have today to go ahead and work through more on it. So I think that's all from my side. I think I can just stop sharing. If there are any questions, I am ready to take it.
Ma'am.
Yes, ma'am. There are a few questions. May I ask, ma'am?
Yes,
Okay.
please.
So somebody asked how to delete duplicate edges from list of genes using Cytoscape.
So once you create the networks, if there are overlaps edges, it will automatically read them and it will merge them into a single node only. So when you put the data through your if you are fetching the data, you can just fetch the data either through public repositories. Once the data is present in the public repository, I just showed you the interface also. It is a simple process to even delete it. If you don't want it, you can just delete the node and then it will not exist in your network. So duplicated nodes will not appear in your network. Once you've built them, Cytoscape will see, and if there are any duplications in your network, either it will merge it all together if you ask it to. So when you click on the option of merge networks, which is a primary feature on Cytoscape, you will get your problem will be solved.
Thank you, ma'am. Somebody has another question, ma'am.
Uh,
Ma'am, there is a question like how to overlap the networks in Cytoscape.
so there is an option called as merge networks in Cytoscape. If you try that, you can merge two networks together. So anything that you do in Cytoscape will require there are two things that will be helping. So you go to tools and you go to merge and then networks, and then you have to select both the networks from your working window, and then probably you can do that. I can share and show you. Okay, so I don't have multiple networks with me right now, but I think if you click on tools, merge, networks, you need to select the network. Right now, I only have one network, so it will not merge anything. You can click on union, intersection, or difference. Union will merge them, intersection. So this intersection will remove your problem of duplicate edges also. It will intersect them, and difference will highlight those which are different from each other.
Ma'am, there is a follow-up question on merge network. Like
Mhm.
if we merge networks in Cytoscape, how can we validate newly formed edges?
Okay. So it is a computational procedure. I hope you understand. So whenever you're trying to build any network, you know, that is why I gave you so much background information that where are these nodes and edges coming from. So if you take the nodes and edges from a curated database, that is a database such as biosites or metasites, which already has curation that is happening behind it, you don't need to validate the edges because of obvious reasons, that you are taking the data from a resource which is already validated through literatures or papers. Now once you've done this, you will have to go back and check about the node, whether there is any reference in the literature which shows that yes, these two have a possible connection, either in any of the database or through the literature that you have. I hope that answers your query.
Thank you, ma'am. Next, we have a question, ma'am. What is the confidence score between before we open the network between different genes?
What is the
Confidence score before we open the network between different genes.
confidence score?
Yeah.
Okay, so confidence score comes from, again, it will not come, you can calculate the clustering coefficient. There is an option, again, I don't know my screen is there or not. There is an option called network analyzer. So now network analyzer will give you the scores that you're talking about. So that will answer your question. And within this period of 1 hour, it is not possible for me to show you everything that happens in Cytoscape. But yes, I can answer the question by saying that you can do network analysis. That's the primary thing you do with any biological network, that you need to calculate the node degrees, clustering coefficient, or and the score, confidence score that you are saying. In terms of networks, that is the node degree and the clustering coefficient. Whether it is behaving as a biological network or not, you need to find out, and then only you will start your analysis.
Ma'am, there is a question. What is the app that we use for identification of disease network?
Okay, so you can use see, any specific problem that you have can be dealt with a number of apps. There are many apps. Like if I want to use expression data, I can use the app called as Experisence. I can use other apps that are based on that on the information. So if you are trying to work through any network that you are talking about, that I'm sorry, I missed it. What was the question? Which disease
The
networks?
app used
Okay,
for
okay, okay.
identification of
Okay,
disease
so
network.
if I was working on Cytoscape and I wanted to find out any kind of disease network or disease module or hub genes, I would have gone for either MCode or for CytoHubba. Both these plugins would give me the hub genes. These hub genes would probably be the biomarker of the diseases, and I can find out more information once I do the gene ontology.
The final question of the day. Can we change M-code score manually after analysis to include more genes in top 2 subcluster?
You cannot change the score after you run the analysis. That is not possible. But you can change, as I showed you in my PPT also, you can change the settings before you run the M code. Okay, so once you are trying to run, you can change if your clusters are too big and you want to make them a little smaller, or you want them more specific, you can change the degree of the nodes that you want in the network, but you have to do it before you run, or you can rerun it. If you are not getting a proper result in the first go, please rerun it and you will get it in the second go according to your data that you are trying to pitch.
I'm sorry to bother. There is a final question from a candidate. Can we study residue interactions in protein using any app in Cytoscape?
I have not done that myself, but I'm sure you can, because if you go through the Cytoscape website and you go through the plugins that are available there is a list of plugins which are available for different topics and headings that will give you your answer. Just go there, check the heading, and what kind of residue interaction are you talking. You can't be doing you can't be docking, or you can't, if you're talking about interaction between, as in docking, or if you're talking about simulation studies, that is not possible in Cytoscape, because it is not a simulation software. It is an interaction and analysis software. So what you can identify is how these residues are interacting with other protein residues. That is probably possible, but individual protein, if you want to see the residues, how is it interacting with the probable drug candidate, that is something which is not possible through Cytoscape.
Yeah, like I will add on to the point what ma'am said. Like you can't talk about protein and the ligand interaction, but you can
That's
talk
not
about
possible.
protein-protein interaction and intra-protein residual interaction that you can see in Cytoscape. That is possible.
You can't be doing docking or
Yeah,
protein ligand interaction in
but you can
Cytoscape.
use intra-protein and inter-protein interactions using Cytoscape, either in terms of energy or in the terms of whatever the interaction, whether it's going to be a hydrogen bond or a hydrophobic interaction. In that kind of terms, you can check out the interactions.
I think you can try out.
Yeah, you can try. There is many modules like Kunan. I think Kunan was being stopped, and now we have Senens, I guess so. So you can try out that module for your protein interactions, residual interactions.
Hello, ma'am.
Yes.
Yes, ma'am. One question by my side. Ma'am, if we want to compound pathway interaction, then how to import data file for compounder pathway interaction?
You're talking about compound pathway interaction. So you
Yes,
will be
yes,
having a list of compounds with yourself.
yes, ma'am.
Okay. So you need to go to either of the databases I listed. You can go to KEGG compound database.
Okay, ma'am.
Find out how your compound works, which are the possible interactions of your compound, and from there either you can draw it in Cytoscape, or you can fetch it through the import option that I showed you.
Okay, ma'am. Okay, ma'am. Thank you, ma'am.
Yeah, welcome.
Hello, ma'am. Can you hear me?
Yes, I can hear you.
Okay, so actually, I have a question, and already I've asked, but still I'm not satisfied with the like explanation. So could you please clear with me? Like, I am working on a project that is identification of anti-cancer drug pathway through drug-drug and gene-gene interaction to find out the most important gene involved in the pathway to deal with the actual cause of the side effect beyond the beneficent effect of drug. So, ma'am, for this project, actually, what I have done, I have found it. First of all, I've selected few drugs, and then I found out the few genes which is associated with that drug. Then, in the previous research paper which I followed it, there is mentioned that remove the duplicate edges and then again go for the analysis of the interaction, but not from the cytoscape. They just mentioned that only from cytoscape remove the duplicate edges. So but I am not understanding how to remove the duplicate edges, and if we are deleting from cytoscape, then why we are not visualizing from cytoscape, why we are using any other tool? So can you please clear with this thing?
There is no need to use other tool. You can do it in Cytoscape only. The reason that they might have used the other tool would have been that they must not be getting what they want from Cytoscape if they were working on it, or probably they did not try it. But as I told you, if you have two networks, or if you have the same network and you run the intersection module, you will get a removal of duplicates by itself. That's one thing that you can try working on. Or else, if you have a representation that you make by yourself, because it's a smaller network and it is an experimental network. So like when I did my PhD and I worked on Cytoscape, I had created the network myself because it was a literature curated network. It was not fetched from string or anything, because the confidence score coming from string is a little less. So if you do that, I had managed with the duplicate links like that only. And what do you call as a duplicate link is, you have a gene A which is getting probably modulated by gene B, but that gene A is also getting modulated by gene X. So that gene A should be present only once in the network. I hope that is what you are talking about as duplicate links.
Yes, yes. And let's say if someone does not remove the duplicate edges and all that, so will it show any variation in the result? Or if I will go through without the removing, so does it
if
the
you
result
if
should be
you
correct or not?
add if you add any kind of expression data or any other experimental data and integrate into your network, it will give. And when you represent it, there will always be a question with your paper that why have you not combined them together into one. So that is why it is important that you combine it. If not automated way, you do it manually. That is my suggestion to your research work. If it's a small network, possibly of 100 nodes or less, please go ahead and do it manually. That will solve your problem in a better way. Don't look out for any automated process, because that may exclude what you're looking for.
Yes, ma'am, got it.
Okay. And if in case there is any other, you can directly get back. Probably that will be easier, because unless I see it, it is not possible for me to comment
Yeah, you
more.
can. The participants who are having like a major doubts in their projects or concerned with their PhD, they can mail this NyBerMan. We can contact the concerned professor Priyaka Narenth will mail you the respective answers.
Yeah, so that way it will be better, because it is
Because
not
it's
possible.
for a single candidate, it's going on for a lengthy process. So, do we have any more questions?
I don't see any in the chat
If there is
box.
no questions from here, I have questions from YouTube, which states that um, the candidate asked, how do we can look into protein ligand interaction?
I think we've already answered that. You can create a network for protein ligand interaction using the same kind of process that we did for protein-protein interaction. You need to find out the ligand identifier from a proper database and then fetch it through Cytoscape and do it. But you can't be doing interaction in terms if you are talking about individual interaction or you want to dock them together or find out that kind of an interaction. That is not possible through Cytoscape.
Okay, there is another question. How frequently data or network updated in Cytoscape?
There are no networks in Cytoscape. It is a network visualization software. So there are no networks that are inbuilt in Cytoscape. That's not there. What networks are present are again already published networks that are available with Cytoscape. So obviously they are validated because they are published and people have already worked on that. So Cytoscape is not a network database. It is a network visualization software. I hope that
Okay, there is
answers.
another one question. Can we look for primer network in Cytoscape while doing recombinant DNA technology?
Again, answer is the same. There is no specific primer network, ligand network, or any kind of network that you're talking about. The analysis will depend on what you want to do with that primer. But for the beginning, you will have to use a tool like primer 3 to get the primer, to get the sequence, or to get the ID for that primer. And once you get that, you can use a network representation to solve your further problems. But it is not possible to have a ligand network or an interaction that you can directly see in Cytoscape. So Cytoscape is a visualization software for protein-protein interactions, primarily gene-protein interactions, metabolic interactions, and then there are plugins which help you analyze that. So if you ask me, probably I can look back and see if there's anything that can work through primer analysis, but primer network is, it works the same way. You need to find out the basic ID or the sequence to get the network built in Cytoscape. I think that answers the query. Is there any further question? Yeah.
If there is any further question, please comment. Otherwise, we would like to end the question session. It seems like there is no any other further question. Ma'am, one last question. What is your role in NyBerMan Bioinformatics, and how can we approach you, ma'am?
Okay, so I'm working as I'm here as an advisor and a tutor, and there are courses which I think are there on the screen now. You can take up a course of R in genomics, which is again a very interesting course where you will be taught from basics to advanced R, and also gene expression analysis or microarray analysis using R. And then we can also discuss individual problems and if you have anything during these sessions, and also an expanded course on Cytoscape, which can be there where what I showed you probably you can have a hand holding of how do you do it actually with a case study. And then there is gene ontology, there is network analysis which can be done for hub gene analysis, which a student was asking that I want to identify the most important gene. So that is also possible by using the plugin such as CytoHub or something. So that you can do. And gene ontology is another interesting part which you can work out, where you can find out the molecular function, biological processes, and the cellular compartments. So all these things can be catered. I can take these sessions and I think they will be helpful for you. So that's all from my side.
Yes, ma'am. Thank you, ma'am. So thank you for this very informative session, ma'am.
Thank you so much for inviting me. I'm glad to be here.
Yeah. So this session was very informative, and thank you for the patience from the students and the participating members side, and thank you for these interesting questions. So your feedback forms are given in the chat room, and internship and the mini courses that were shown in the screen. Their links were also shared in the chat room. Please check out. And to have many contents like this and to view many webinars like this, you can follow LLB Scientifically in YouTube. Thank you. Thank you, ma'am.
Thank you so much. I'll take your leave. Thank you.
Thank you, ma'am.